Okay, so we've done neural pathways, and we've talked about the basic overview of the nervous system, having the central nervous system and the peripheral nervous system. But what is the nervous system made of? Well, it's actually made of two types of cells. There is the neuroglia, and then there's a neuron. So the neuron they're the star of the show. So I'm going to draw a star. Those are the star of the show. That's what everybody looks at, everybody works on, everybody looks at these. The supporting cast and the people who set up the stage and keep things running. So this is all the so this is all the roadies, help set up the stage for the big rock star. And so those are called neuroglia. So you have neuron cells and neuroglial cells, or neuroglia. So we have neuroglial cells, or neuroglia. So, and the neuroglia support the neurons. They protect and furnish nutrients to the neurons, and they also help increase the speed of the transmission of a nerve impulse. And we're going to see how that works here in a second. Next slide. So here's a photomicrograph of nervous tissue, some motor neuron. So you have the cell body, so that's in the middle here. That's the big fat part. So this is the cell body here. Okay, and then you have dendrites. Okay, and a neuron, and we usually use a motor neuron as a prime example, just because they're kind of your typical neurons, and that's what a lot of your neurons are. Motor neuron means it's producing some sort of effect, so it's generally in the afferent pathway. And a neuron can have a dendrite or many dendrites, and they usually have many. Okay, and so you have the cell body, and then you have dendrites. So here's a dendrite, here's a dendrite, here's a dendrite, here's a dendrite, here's a dendrite coming off. So this guy has 5 dendrites coming off of him. Each neuron only has one axon. Oops. So this is your axon here. I'm just going to circle it. So you have one axon per neuron. And where we're also going to talk about where the part where it attaches to the cell body is an important part. We're going to talk about that here in a second. You also have neuroglial cells surrounding this neuron. So all these little dots, here's one, here's one, here's one, here's one. All those little dots, those are all the nuclei of neuroglial cells. So now you can see there's a whole bunch of these. There's one, there's one, there's one, there's one, all over the place. There's all these different neuroglial cells that are providing support for the neuron and helping maintain its environment, keep it supported, hold it in place, do different things for it. Next slide. So here's another picture of neurons. So there's three neurons here. Here's one, here's 2, and here's 3. Okay. Then you have an extracellular matrix that's been produced outside these neurons, holding them all in place. So you can see the extracellular matrix that's pointed to here. And then you have nuclei of the neuroglial cells. So there's one there. Here's one here. Here's one here. There's one there. There's one there. One there. One there. They're all over the place. There's a tons of these neuroglial cells all over the place. And they're also providing support for producing an extracellular matrix, providing different a nice environment for the neurons to function. So, and then you have dendrites on each of the axons. So here's some of the dendrites here and here, and then you have an axon. So here's an axon coming off of this guy here. So you can see the axon. Okay, next slide. So, structure of nervous tissue, you have neuroglia, which are cells that support, protect, and furnish nutrients to neurons and augment the speed of neuron transmission. They're smaller, and there's lots more of them than there are neurons. They do not generate action potential, so they do not make no nerve impulses. But without them, the neurons won't be able to make nerve impulses or transmit them very well because they need this whole supporting cast to help them out. So you have your neuroglia, and see, that looks like an S. It's actually an O. So anyway, all right. The neuroglia, there are 6 types that are found. There's 4 in the central nervous system, and there's 2 in the peripheral nervous system. In the central nervous system, you have astrocytes, oligodendrocytes, microglia, and ependymal cells. Astrocytes have many processes. They look star-shaped. They have what are called perivascular feet. Peri means around, vascular means vessel. So it goes, the astrocytes go around things, and they help form the blood-brain barrier. I remember the name. I'll go on with these. The oligodendrocytes, they form myelin sheaths, and we'll talk about what a myelin sheath is, and they support the central nervous system neurons. The ependymal cells, they line the central nervous system. They have cilia, and they form the cerebral spinal fluid that's in the central nervous system. The peripheral nervous system has 2 types of neuroglial cells. They have Schwann cells and they have satellite cells. So we have the Schwann cells. If I can find it down here, where am I? There are Schwann cells and satellite cells. So the Schwann cells wrap around the axons of the neurons in the peripheral nervous system, and the satellite cells surround the neuron cell bodies. So the satellite cells help the neuron cell bodies, they help their environment, and the Schwann cells wrap around the axons and help transmit, help increase the speed of nerve impulses. The astrocytes, I usually refer to those. I've a friend named Sarah from years back, and she tells a story about when she was little. She would go into a store, and Sarah has an issue. She touches like everything, so she would touch everything in the store, and then her mom would always tell her, Sarah, don't touch all this stuff in the store. And she would say, Sarah, assume the position. So Sarah had to go and put her arms across her chest. So I call these Sarah cells because that helps me remember them, because they go out and they have many processes. They go out and they touch everything all over the place. So that's what astrocytes are. Astrocytes are the Sarah cells to me. Anyway, just a little side story there. Okay, next slide. Okay, here we have the neuroglia of the central nervous system. And so here's the astrocyte. That's the Sarah cell. And you can see this astrocyte, there's the astrocyte cell body. And it has all these processes sticking out. And it reaches out and it touches like everything around it, everything it can find. Astro meaning star, so it's like a star. And this is a capillary going through the brain here. This is a neuron cell body here. This is an axon coming down here. So you can see the astrocytes reaching out and touching not only the capillary but also the axon of this neuron here. It's also reaching out and touching the cell body of an oligodendrocyte over here. And it's also reaching out and touching the axon of another neuron right here. So here's another neuron. Then you have, it helps anchor neurons and blood vessels in place, is one of the big functions it has. It also regulates extracellular environment and also facilitates formation of the blood-brain barrier and also helps repair damaged tissue. The oligodendrocyte, that helps myelinate certain axons in the central nervous system. So what they do is, here's the oligodendrocyte cell body, and it has a process sticking off here, and you notice it goes and wraps around the axon that's running underneath it. And so here's another process, and it goes and wraps around this part of the axon. And it reaches out here and goes and wraps around this. I wish I would quit doing that. Good grief. So I can't draw slowly with this apparently. So it wraps around an axon of more than one neuron. So it doesn't have to. It's not exclusively wrapping around only one neuron. It's reaching out and touching other neurons and wrapping around them and producing what's called a myelin sheath. So And then we're going to talk about what a myelin sheath is, and so that's why it says it myelinates certain axons. Then you also have microglial cells, so they're little small cells. And so here's the neuron, here's the cell body, and they also have little processes sticking off of them as well. And they go out, if there's little particles that occur in the brain, they go and they eat them. So they act as phagocytes. So phagocyte meaning a cell that eats things. Then you have ependymal cells, and ependymal cells are these kind of cuboidal cells. So what type of cells are cuboidal? That would be epithelial cells. So these are epithelial cells because there is space right here. Over here, there's fluid that's being secreted into the space. You have cilia all through here. Okay. So and that would be your nucleus right there. So they line cavities, and that's what you expect for epithelial cell. They have cilia that circulates fluid around the brain and spinal cord, and some of these cells secrete this fluid. And one of the things they really do is they process fluid coming out of the capillary over here, and the fluid gets processed and made into this the fluid that's in this space. So this is a space. Let's see if I can write space, and the space has cerebrospinal fluid. That's supposed to be an F. So let's see if I can write that word out. So cerebrospinal fluid. So the ependymal cells are important for. They line the cavity of the cavities that are in the brain. We're going to brain and spinal cord. We're going to see those later on in the next section. So, next week. Next slide. So here's another picture of neuroglia of the central nervous system. So we have an oligodendrocyte, we have a microglial cell, we have an astrocyte, we have an ependymal cell. So just showing you where these cells are again. So here's your microglial cell. Here's an oligodendrocyte here with all these processes sticking off, touching everybody. And then we have, and here the yellow, these are neurons here and here and there. Astrocytes, they're reaching out, touching like absolutely everything. The oligodendrocytes is reaching out here, touching axons. And then the ependymal cells, which are lining the cavity. So there's cerebrospinal fluid down here. So this is all your different types of neuroglia in the central nervous system. You have oligodendrocytes, microglial cells, astrocytes, and ependymal cells. Next slide. Then you have neuroglial cells of the peripheral nervous system. So peripheral nervous system. Okay. So you have satellite cells and you have Schwann cells. Satellite cells wrap around the neuron cell body. So all the satellite cells are all up around here, and they're just nourishing that cell body and keeping its environment around it so it's for something it likes, and keeping basically homeostasis around the neuron cell body. Then we have the axon down here, and that's what the yellow is, and wrapped around it is the Schwann cell, and you can see it's wrapped around, and there's multiple layers around this thing. And then this part right here is the nucleus of the Schwann cell. So what happens is the cell wraps around and around and around and around the axon. So here's how a Schwann cell works. What we're going to do is I got a pencil here, yellow, so that's representing the axon. And then I have a plastic bag with a little part of a keychain, and it's representing the nucleus of the cell. So the plastic bag is representing the cytoplasm, and that's the nucleus of the cell. So what you have your axon here. It's your axon, and then you have the Schwann cell, and it wraps around all the way around, just and you roll it, roll it, roll it, and then you get this little air in there. So you get this wrapping that's occurred all the way around the Schwann cells wrapped all the way around this axon. I'm going to try to get the pencil out. So you have all these layers now, and so here's a real simple kind of dorky model though of a Schwann cell around an axon, but that's how it works. And so you have these layers and layers and layers. So what's your cell membrane made of? Remember, this is a cell membrane. It wraps around and around and around and around the pencil. Cell membrane is made of lipoprotein. So what's lipid? Lipid is fat, and lipid doesn't conduct electricity very well. So this acts as an insulator around the axon here, which is conducting electrical impulses. So that means electrical impulses don't just like fly off here. It can actually keep those electrical impulses inside the center space here of the axon and keep them from escaping. And then you have the cell nucleus on the outside here. That's on the top side. That's what this little part was down there. And let's see, I'll draw a little arrow to it. So that was your cell nucleus here. It was there. And so this little part here is called the axolemma. So that's your axolemma. So there's your model of a Schwann cell wrapped around an axon. Use a plastic bag and put a little bead or something in there to represent the nucleus and wrap it around and around and around the pencil, and that shows you how it all works. Next slide. Okay, so here's neuroglia of the central nervous system again. This is all I did was blow up the one of the previous ones. I just wanted to show you a couple little things on this. The astrocytes, they have all those different processes. They wrap around and cover neurons and blood vessels and keep the neurons in place. Okay, that's one big thing to do. But they also guide neurons during development. They control the composition of the chemical environment of the neurons. They help form the blood-brain barrier. So, but to me, the interesting thing is they guide neurons. So neurons, when they're moving around, they get taken to a certain place, helped along by these astrocytes. Next slide. Here's an actual photomicrograph of astrocytes. And so you can see all the different processes that come off of them. They look like little stars. So here's an astrocyte here. Here's an astrocyte here. Here's one here. There's another one here. So just all through. Here's another one here. So all these blobs, all these dark blobs all the way through. Here's a really nice one right here. So anyway, that's astrocytes. Next slide. Here's another photomicrograph of an astrocyte. So you have your blood capillary up here, and you have the cell body of the astrocyte here, and they have these processes going up here and grabbing onto this, grabbing onto the capillary, reaching out up here, and it's reaching out all over the place, trying to grab onto things, hold things in place, as well as help maintain the environment. So I'm drawing around a lot of these processes that's sticking out off of the astrocyte. So anyway, next slide. So oligodendrocytes, this is the one of the two types of cells that produce the myelin sheath, oligodendrocytes, and the other ones are the Schwann cells. Oligodendrocytes produce the myelin sheath in the central nervous system. Schwann cells produce the myelin sheath in the peripheral nervous system. So these produce these myelin sheaths around axon, and that myelin sheath increases the speed of nerve impulses considerably. A signal can move about 400 kilometers per hour. So, and you can see here the oligodendrocyte here, and its processes sticking down and making a sheath around the axon. Now you don't have to make a really great sheath around the axon. Well, here actually it reached up to a different axon up here because it doesn't have to be all on the same axon. But so you can see that from the process coming up here, and then this process going down to this one, and this process going over to this one. So the sheath doesn't have to be as wrapped as many times around the neuroglia in the central nervous system as it does around the peripheral nervous system neurons, because the signal doesn't have to travel as far. It doesn't have to travel as fast inside the central nervous system, inside the brain. So when you're inside the brain, it's a very short distance for the nerve impulse to go. So you need a little bit of insulation there, and that's what the myelin sheath does. It insulates the axon. It's kind of like if you've ever done electrical wiring, you'll have the copper wire on the inside, and you have the rubber or plastic coating on the outside of the wire that provides insulation, so the electrons stay inside the plastic sheath, plastic or rubber sheath, providing insulation on the outside for the outside and keeping the electrons on the inside where the copper wire is. So you don't get a short circuit. So this works exactly the same way. You have these myelin sheaths that keep the electrical impulses inside the axon. So next slide. Okay, here's some diagrams. These are not photographs of the oligodendrocyte. So this is just a drawing, but it's going and showing the oligodendrocyte here, cell body, and all the processes coming off. There's process coming off here, wrapping around, and this process coming off here and wrapping around. And you can have a bunch of layers in here. Doesn't have to be just one layer, but it doesn't have to be a whole lot of layers either. So, and here's another one, oligodendrocyte. It'll reach out and maybe touch a neuron as well here, but it also forms this myelin sheath as well around the axon. Next slide. Here's an actual oligodendrocyte. They stained this. They added an antibody that has a fluorescent molecule on it. Then they shine a light from the side on it, and that molecule fluoresces. And generally, you have red or green when you're doing these kind of fluorescent stains. So this stain happens to be a green stain. So you can see all of the processes coming off of this oligodendrocyte. So there's the cell body there, and then you have all these little stringy little processes coming off, going all over the place. Just reaching out, trying to touch different things. So you'll have the body, and they have all these processes coming off like that. So next slide. Here's a closer view of the little higher magnification of the showing the cell body, this bright part right in the center, and then all the processes sticking off of the cell body. Next slide. Okay, so there's number 3, microglia, microglial cells. Those are the phagocytes. Say if there's cell tissue that dies, they'll eat that up. So necrotic tissue is dead tissue. They'll also go and phagocytize bacteria, viruses, or just general debris that occurs in the brain. So if there's maybe some proteins that escape off the surface of the cell, they'll eat that too and break that down. Next slide. Here's some pictures of microglial cells. You can see that the cell body is here, and they've got these processes sticking off. I'll point down. There's a cell body there, and they got processes sticking off there, and it's hard to see on the black background. Let's see. Let me change my pen color. See if that works. So yeah. So there's a process, and here's the processes sticking off that way, and here's a process sticking off that way off the cell body of the microglial cell. Okay, and next slide. And the ependymal cells, those are the ones that line the central canal and the ventricles of the brain. Those are spaces that are within the brain. You actually have holes in your head, holes in your brain that are fluid filled, and these cells help form the cerebral spinal fluid, the CSF. And their cilia move the cerebral spinal fluid through the ventricles and up and down the central canal of the spinal cord. Next slide. So here's a cross-section through ependymal cells, and you can see they're cuboidal, maybe columnar shaped. And you have the cilia up on top up there. So here's your cilia, and here's your cell, and then there's a membrane along the bottom, and then this is all going to be neural tissue down here. And so these are all satellite cells and other types of neural tissue cells, and maybe some neurons scattered in there somewhere. It's possible that's one there. I can't tell for sure. It could be a capillary too. It's hard to tell on this slide. Anyway, next slide. More ependymal cells. So we have the spinal cord. This is the central canal. And H&E stain means hematoxylin eosin stain. So that's why they're reddish. And here's ependymal cells and the cilia sticking up the top up here. And the cells, that's a nucleus, a nucleus, a nucleus. So there's your layer of ependymal cells. Next slide. Neuroglia of the central nervous system. There they are again. So just to wrap it all up, oligodendrocytes over here, microglial cells, astrocytes, and ependymal cells. Okay, next slide.